Everyone. Today we are going to see Mendelian genetics and human genetics. So there are certain concepts that I want you to understand. So the first of the words that you need to understand is genotype. And when we say genotype, it could mean two things. The first one is when we say genotype, it could mean the entirety of the genetic makeup of an individual. That means all the genes that an individual, or it could say an individual cell has, could mean a genotype, or in some cases it might include the whole population. For example, for human beings, the entirety of the genes that are found within a human being, or in general human beings. Or it could go down to all the alleles of a single gene. For example, uh, let's say you have two or three different alleles, or let's say five, six different alleles that determine the color of the eye. So even though we're talking about a single gene that determines the color of the eye, you might have different alleles. You might have an allele for, let's say, brown eye, or for gray eye, or for blue eye. So that's what genotype is. When we say phenotype, this is something that you can measure. For example, it could be a height, or it could be weight, it could be color. It's something that you can measure, or something that you can observe. So, so phenotype is, you can say, it's a manifestation of your genotype. So genotype is just the pool of alleles of a given gene or multiple genes. And when you say phenotype, it means the physical, or the physical aspect of genotype, something that you can measure, observe. The other thing I want you to understand is Punnett square. I'm sure there should be a Punnett square over here. I think I have a Punnett square somewhere. Yes, this is what a Punnett square is. These are the alleles from one parent, and these are the alleles from the second parent. So when you multiply them G by G, you will get this one. And this chart, or you can say this square, is called a Punnett square. And you should be able to manipulate the Punnett square at this point. Let's say if um, the alleles for the one of the parents is small g's, or it could be one capital and the other small, you should be able to multiply. So these are the alleles that determine the color of the pod. In this case, I think it's a P. So we have this allele, the capital G allele, and we have the small g allele. And the capital G allele, it codes for green color. Uh, the other thing is related to fruit fly. You should be able to differentiate between sexes. You should be able to identify a male or a female fruit fly. They are different in their size, they are different in the shape and color of the abdomen, and they are different in the claspers or sex combs. So, for example, male fruit flies, they tend to be smaller in size, and the shape of the abdomen to be tends to be a little bit rounded instead of pointed, and the color of the abdomen tends to be dark. So they are small, and they have a pointed abdomen, and it tends to be darker, like dark brown or sometimes blackish color. And they have claspers or sex combs, or in a sense they are like a male genitalia. So these claspers or sex combs are found at the end of the abdomen. So that's what one identifying feature for a male fruit fly. If it is female, it's going to be large. The shape is going to be pointed. It's like a letter V, and the color of the abdomen tends to be clear compared to the male fruit fly. The other thing is you should be able to identify fruit flies based whether they are wild type or mutants. I want you to watch a video or just go on Google and differentiate between wild type and mutant fruit flies. So try to see what kind of wing wild type fruit flies have, what is their eye color, what's their body color. When you say wild type, by the way, it means uh, what we can find in the regular population. For example, uh, uh, let's say height in the wild type or in the regular population, uh, tall trees might be very common. That means any tree that's short tends to be a mutant. So when you say wild type, it means a common variety in a given population. So you should be able to identify different kinds of mutants, and I'm sure you have seen this one in one of the virtual lab assignments, I guess. The other concept is monohybrid cross. As the name implies, it is mono. That means the cross is about a single gene because it is a monohybrid cross. Is mono. Second, it is hybrid. Hybrid means there are two alleles. Even though it is a single gene, there are two alleles that you are dealing with. So in monohybrid cross, the parents are heterozygous. That means they have two different alleles, and you are trying to determine the phenotype and genotype of the offsprings based on the alleles from that single gene. This is actually a monohybrid cross because we have two genes that we are dealing with, two alleles, sorry, a single gene, but two alleles. The two alleles are, for example, for one parent, we have only a single allele G because they have two copies of a single gene, or we can say we have only one allele, the same one here. We have only a single allele, capital G. So this is a monohybrid cross because we are dealing with a single gene that determines the color of the pod. If we deal with, for example, whether it is wrinkle or if it is smooth, then we are dealing with a second gene. And when you deal with two genes, we are talking about uh, a dihybrid cross. That means the parents are heterozygous. This one is actually, it's not a monohybrid cross per se because each parent has a single allele, not different alleles. So to be a monohybrid cross in this case, this has to be a capital G and this has to be a small g because the parents have to be heterozygous. In this case, they are not heterozygous, they are homozygous because they have small g's or two capital G's. Now, when we uh, come across with a research in Mendelian genetics, there are certain words that might be a little confusing. I'm not going to test you on this, but I just want you to understand. I just want you to understand the concept behind it. So when we say null hypothesis, for example, in Mendelian genetics, uh, the concept is not that complicated. For example, let's say you are trying to see if a certain chemical determines the height of a certain plant. So then you have two options to phrase your hypothesis. One you can say what you can one option to say is, for example, the chemical will not have any influence on the height of the plant. That means you are saying there is no difference between the plants that receive that chemical and the plants that didn't receive in terms of height. So you are saying there's no difference. Or the other option is you can say the chemical is gonna influence the height of the plant, or in this case, you can choose two ways. You can say it's gonna increase the height, or it can say it's gonna decrease the height. So this is like, if this is one form of alternate hypothesis. You are not saying, oh, it's not going to influence it. You are actually saying it is going to influence it. On it depends what you are going. Either it's going to increase it or it's going to decrease it. So null hypothesis is when you choose the no difference route. That means you are saying there is no difference between the one that received the independent variable and the one that didn't receive the independent variable. So in research, for example, you might say, uh, uh, there is no difference in the color of the plants or something like that. So that's what a null hypothesis is. A chi-square is a test that we usually use in Mendelian genetics. So it deals with frequency. For example, you are trying to determine the color of offsprings after you do a monohybrid cross or a dihybrid cross, and then you are trying to count the different colors. So in chi-square, you are trying to compare the expected frequencies with the actual frequencies or the observed frequencies. That means chi-square is a test of difference between observed and expected frequencies. And when you talk about degrees of freedom, uh, in genetics, you just try to determine how many phenotypes of a single gene you have. After you determine the number of phenotypes, you subtract one, and that gives you the degrees of freedom. There is a lot of science behind it, but you don't need to know that, at least for this class. So that's all regarding Mendelian genetics. Um, the human genetics, there are certain concepts I want you to understand. The first one is karyotype. Uh, this is what a karyotype is. It shows you all the chromosomes we have as a human being. Uh, Chromosome 1, 2, 3, 4, 5 is showing you the size, and they are always in pairs, one from the father and one from the mother. And this one is especially a male karyotype. How do I know that? It says X and Y at the end. Only males have X and Y genes. So this is a male karyotype. If it was a female, the females have two X's, that they have two copies of the X chromosome. So if it was XX, we can say, oh, this is the female karyotype. So a karyotype is, you can say it is a chart or an image that shows all the chromosomes of a human being with their relative size, and yeah, with their relative size. Genes, human genes, we can classify them into two. You can say sex-linked genes and non-sex-linked genes. The sex-linked genes are X and Y. These are the sex-linked genes, or the X chromosome and Y chromosome. We call them sex-linked genes. That means this is a chromosome, so there are genes within this chromosome at different segments over here too. And as you can see, Y tends to have less genes compared to X. So these are sex-linked genes, X and Y. So yeah. The non-sex linked genes are all the 22 pairs of chromosomes, and these 22 pairs of chromosomes, we call them autosomes. So these are what autosomes are and what sex-linked genes are. Alleles. We have two kinds of alleles. Alleles are, by the way, they are varieties of a gene. For example, a gene for height. It might show, you might be tall, you might be short. So okay. So you might be tall or you might be short. That depends on what kind of allele you have. For example, in this case, the small g is called the recessive allele, and capital G is called the dominant allele. The reason is whenever there is the dominant allele, all the other alleles will be masked. In this case, when there is G, the color is not going to be yellow because there is G. So wherever there is G, the the other alleles will be masked, and the G is going to overshadow the rest of the alleles. So dominant allele, this one is like its phenotypic effects are going to be expressed. If it is recessive allele, the phenotypic effects are going to be masked unless you have two copies of that recessive allele. The other thing I want you to know is chromosome structure. There are two concepts that sometimes confuse students: the homologous chromosomes and sister chromatids. Um, I'm going to go to this junction. Maybe that could explain things. Okay, when we say homologous chromosomes, let's say this one is a sex cell. Let's say this is from this chromosome X and Y, and then they make copies, and you are trying to separate them. So before this happened, uh, there you there were like in this case there should be only two chromosomes in this case. So these chromosomes, one from the father, one from the mother, they are going to be duplicated. That means the father's chromosome is going to be duplicated. Now you have one chromosome, not just one, but you will have two copies of the same chromosome, but still it is called one chromosome. And for the mother, the same. There was one chromosome, and that's going to be duplicated. So now you have two. We don't say two chromosomes, but it's like you have two copies of that single chromosome. Now that means you have two from the father, two from the mother, and they are going to be close to each other. And these are called homologous chromosomes. Homologous chromosomes means the chromosomes themselves plus their copies. That means that they're like in tetrads, like in groups of four. And these are called homologous chromosomes. So let's say this is meiosis one, or let's say the cell is dividing. So let's say, no, actually that's the topic we're going to cover later. But these are called homologous chromosomes. Now, usually homologous chromosomes separate. In this case, for example, this is from the father, this is from the mother, let's say. This is going to go to this side, this is going to go to the mother's side. Now we are saying the homologous chromosomes are separated. Now, these are the same copies and they are going to separate. And these two are called sister chromatids. So the homologous chromosomes come before the sister chromatids. And in meiosis one, it is the homologous chromosomes that are going to separate. And in meiosis two, it is the sister chromatids that are going to separate. These are the sister chromatids, they are exact copies. Homologous chromosomes, one from the father, one from the mother, plus their copies. That means you are going to have four. So homologous chromosomes, they separate during meiosis one. And if disjunction occurs, it is going to be very severe. This is the one I want to talk about. So let's say if these two chromosomes, instead of taking like one copy here, one copy here, like the one on the left side, if both copies are going to go this side, that means at this stage, the homologous chromosomes, they were supposed to separate over here, but now they just separate over here. And as I said earlier, this is the place where sister chromatids were. So now in meiosis two, instead of going forward with uh, what like a single chromosome, you are going to have two chromosomes, which is not right. So this is very severe because these, they don't have anything, and the daughter cells, they don't have anything. Here They were supposed to get one only. Now they have two. So in this case, it is very severe. Over here, that means when the homologous chromosomes separate fine. But at this stage, these sister chromatids, they didn't separate. So two copies are going to go here, and this one is not going to get anything. On this side, at least they get at least one copy. So this is good. This side, at least 50% of the offsprings are fine. But the other 50%, they are not fine. One is getting something, the other one is not getting something. Over here, none of them are normal. Over here, at least 50% are normal. 25% they lack a chromosome. Another 25% gets an extra chromosome. Okay, so sister chromatids, if they fail to separate, at least it is less severe. By the way, when you say disjunction is a failure to a failure for chromosomes to separate from one another. It could be sister chromatids or it could be homologous chromosomes. Meiosis is the production of gametes. So you start from a diploid cell and you end up having a haploid cell. In mitosis, you start from the diploid cell and you end up having a diploid cell because in mitosis you just duplicate the chromosomes and then you separate it in half. So you are going to gain back what you had already. So the daughter cells and the parent cell are exactly the same. For meiosis, the daughter cells are completely different from the parent cells. There are certain words that might be confusing to some of you, but I'm sure most of you know these. So when it comes to heterozygosity of alleles, there are certain words that we use to describe it. For example, when we say homozygous, we are saying that person has the same alleles of a certain gene of interest. For example, it could be TT or it could be TT. If T, let's say, is an allele that determines the height, when you say TT, it's called homozygous dominant. And when it is TT, it's called homozygous recessive. And in some cases, you might have two different alleles. One is for height, like you are going to be tall. The other one is going to say, oh, you are going to be short. So when you have two different alleles, it's called heterozygous, or you are called heterozygous to that gene of interest. When you say hemizygous, it means you have only one copy. You either have a T or a small t. You don't have both copies. That's hemizygous. Uh, I'm not going to test you on pedigree, but I want you to uh, at least I want you to read a pedigree chart. So there are certain things I want you to understand when it comes to pedigree. So when you see a circle, it means this is female. When you see a square, this means this is a male. So female, male, circles for female, and squares for males. And you might have shades or you might not have shades. For example, this one is shaded. These are all shaded. So when you say they are shaded, you are saying they have the recessive allele or they are affected. Not just they have the recessive allele, they are actually homozygous recessive. In this case, they are affected means they have the recessive gene. So shades means they are affected already. That means they have both copies of the recessive allele. When you have when you have no shades, it means you have the dominant allele. You might have the recessive allele too, but because of the dominant allele, you are not showing the recessive gene. So when you say no shades, it means you have the dominant allele. When you have shades, that means you have both copies of the recessive allele, and because of that, you are affected. When you see a cross, that means that person is deceased or passed away. And there are three levels: first level, second level, and third level. First level is the generation of parents or this is generation one, or you can say parents. This is generation two, or this is for children, and generation three, which is for grandchildren. And you can use this information to determine, for example, if you know the genotype of the grandchild, you might be able to determine the genotype or the phenotype of the parents. Sometimes, if this wasn't given to you, you could actually backtrack and determine the genotype of the children or the parents. Or in some cases, you could go downward. So this is just for information purpose. I think that's what I have. Yep.